All right, we are recording. So, Brian is here. He is here, right? No, that's not great. He is not here. Darby is here. Hamilton Fisher is not here. Hamilton is here. Hellman is online. Ashley is here. Crystal is online. Taylor is here. George Rollins. Saunders is online. Shamlin. Caleb Pierce. Vargas. Gunn. And Curry is here. Let's see. Did I miss anybody?
Okay.
All right, everyone's muted now. That's good. Okay. So, yeah, everybody's accounted for. So before we start recording, I got a question about when the exam is, and it really does depend on when we finish the next chapter. The next chapter is actually pretty quick, so it is possible the exam will be on Friday, but we'll have to play it by ear. Let's see, we have Fisher online. Speaking of online people, don't forget to send me the attendance words. 5 minutes, at least 5 minutes after we get finished. Yeah, so anyway, today we should be able to finish the respiration chapter and we can jump into the photosynthesis chapter. So do we have any questions about anything else before we get started? All right, so this is where we left off. We actually finished talking about respiration, cellular respiration, aerobic cellular respiration. And then we started jumping into fermentation. We did discuss fermentation yet, right? I think mentioned it and didn't get into it. Right. All right, so anyway, does anybody remember what is the big thing that we're after with cellular respiration? Quick review of the stuff we've already said. What are we after? What is the purpose of cellular respiration? What molecule are we trying to make? And I said for the last chapter, if you remember anything else, remember this one sentence. So what are we after? ATP. Perfect. Thank you. ATP is what we're after. Yes. ATP. And that's still the same situation with fermentation. And I know we did talk about learning objectives. Yeah, that's exactly where that thought, if I remember correctly. So, yeah, because I asked if any of you held your breath before, asked how many of you died when you did it, right? And you didn't die because you can hold your breath a little bit because your cells can make ATP without oxygen for a little bit, and that is what fermentation is. I'm probably not going to ask you the definition of fermentation. Just like cellular respiration, we need to know the 3 different stages. It's kind of the same situation for fermentation. You just need to know what fermentation produces other than ATP. So let's talk about it. Here's a number you might need to know, and I actually think that would be the first attendance word or number, if you will, 15. Actually, no, I'm not going to ask you that. But anyway, after functioning anaerobically for about 15 seconds, your muscle cells generate ATP by fermentation. So again, without oxygen, after a while, your cells make ATP in a different way, which makes sense. And how many of you have ever run or did something to where you were out of breath? Right? A lot of us, I know I have. That's basically the same thing. I mean, not completely without oxygen, but when you're out of breath, that means your cells are using more oxygen, generally speaking, than you're giving them, right? So that's where this kicks in. What you do need to know about fermentation, there's 2 different types of fermentation, but whichever one we talk about, you need to know this. Fermentation relies on glycolysis. So of the 3 stages, we only do glycolysis for fermentation. Reason being is because glycolysis is the one of the 3 stages that doesn't directly require oxygen. We're going to talk about how that works here in a second. And as I mentioned before, it nets 2 ATP per glucose molecule. But the most important thing, I'm going to even put it next to that because I'm not going to ask you those questions. But just remember, if we're talking about fermentation, either type, because I'm about to tell you those 2 different types, either type, it's all about glycolysis. So any questions about that? All right, so if you remember, when we were talking about cellular respiration, aerobic cellular respiration, one with the 3 stages. The whole purpose there, right, we were stripping away high energy electrons, right? We broke down glucose, we stripped away high energy electrons, and then those high energy electrons were used in the electron transport chain to produce a bunch of ATP, right? So we're still kind of doing the same thing. We still want to strip away high energy electrons. So we still need NAD+, because remember, when it doesn't have the electron, when it's ready to accept the electron, it's called NAD+, and technically it's called NADH once it has the electrons. We still need the same thing. We still need NAD+. We need to strip away the high energy electrons. And in aerobic conditions, that's not a problem, right? Because we strip away the high energy electrons. NADH takes it to the electron transport chains. They dump them off in the mitochondria. And then you have NAD+ ready to go again, right? So you keep stripping away those electrons. But that is not what happens when you don't have oxygen, right? Without the oxygen, you have nowhere to send those electrons, and that's the problem. Any questions about the slide before I show you kind of a picture to remind you what I'm talking about? All right, so this is the picture from cellular respiration, right? So again, we're stripping away high energy electrons. Those high energy electrons are taken to the electron transport chain, where they go down, from protein to protein, until finally oxygen accepts them, and then that's the process that produces a bunch of ATP. But again, here in fermentation, we don't have the oxygen. So here we have these high energy electrons, we have the NADH, and nowhere to put the electrons. And without having those NAD+, right, without having this thing ready to strip away the electrons, then glycolysis just is not going to work. So that's where we get this. Basically, your cells recycle the NAD+. They cannot occur under anaerobic conditions. So again, like I just said, see everything I just said in writing, there's no oxygen to accept those electrons. So instead, this is what you need to know, those electrons are added back to the pyruvate. So remember, in glycolysis, you have the glucose molecule and you break it down into pyruvate. Remember, that's basically what happens in glycolysis. So here, instead of those electrons being taken to the electron transport chain, since they have nowhere to go because there's no oxygen, they just put them back onto the pyruvate. And then again, you have the NAD+, it's ready to go again. And that keeps glycolysis working. And another thing you need to know, this is probably the bigger, almost definitely going to be a question, it produces lactic acid. So fermentation in human muscle cells or in any of your cells produces lactic acid. You need to know that. There's 2 different types of fermentation. The one that happens in your cells produces lactic acid. That would definitely be a test question. And then yes, your book points out that that lactic acid is then transported to the liver, and the liver then converts it back to pyruvate. But I'm not going to ask you that. This is the bullet point. As far as the exam is concerned, that's the most important bullet point on the slide. Is that you produce lactic acid. Your cells produce lactic acid. Any questions about that? The next slide I'm going to show you is just a picture of this process that shows the process of breaking down that glucose to form pyruvate, stripping away the high energy electrons, and then the high energy electrons having nowhere else to go, so they're added back to the pyruvate, and that produces lactic acid, and there's the picture. So again, you have the glucose to start with. We break it down into pyruvate. We strip away high energy electrons and produce a little bit of ATP. Those high energy electrons usually go to oxygen, basically, they can't hear. So instead, they're added back to the pyruvate, and now you have lactic acid. Any questions? All right. If you download this PowerPoint, you can watch this little animation that shows a different type of fermentation that we're about to talk about. So it's everything I just said is just animated. So your book points out that fermentation can sustain a lot of microorganisms. Like for us, it doesn't work, right? Yes, we can do it for a little bit. We get lactic acid. That's why you get sore from working out, or one of the reasons why you can get sore from working out, right? Because you added oxygen. So you form or you make lactic acid in your muscles, right? But for microorganisms, they can live that way, right? If you're like a single cell organism, you don't necessarily need the whole cellular respiration. You might be able to live off of fermentation. I'm not going to ask you this, but your old book pointed this out, so I kept it. Lactic acid from yeast, right? So we use yeast to make things like cheese, sour cream, yogurt, soy sauce, pickles, cabbage, olives, sausage, meat products like pepperoni, things like that. You can look that up if you want. There's other stuff too. Like what other stuff do we use fermentation to make? If you want to look that up for an independent word. But yeah, as far as the exam is concerned, that's not an important slide. This isn't necessarily either. I'm not going to ask you any questions about yeast, but let's talk about it really quickly. Yeast is an interesting organism, as far as I'm concerned, because it is single-celled, but it's not like a bacteria. It is a eukaryote, so it has mitochondria, and it has all the organelles, but it's single-celled. So because of that, it is actually capable of cellular respiration and fermentation, right? So yeast is interesting. You can do cellular respiration, and it can do either of the types of fermentation. So it can do the one we talked about previously, which makes lactic acid, but it can also do this. And this is another thing you need to know. The other type of fermentation, as you probably already do know, produces alcohol. Yes, technically it's ethyl alcohol, aka ethanol. And I'm not going to get that specific. It's not like you're going to have a multiple choice question whether is it ethanol or is it isopropyl alcohol, right? Just know that the other type of fermentation produces alcohol. And then, yeah, your book points out that that's obviously used to produce beer, wine, liquor, and stuff like that. But another thing you need to know is this little bullet point here. When producing alcohol, this type of fermentation, it also produces carbon dioxide. And yes, technically that causes bread to odorize, but I'm not going to ask you that. This is not a culinary class. So again, big picture. There's two different types of fermentation. One of them, one that you guys do, the one that we do in human cells, produces lactic acid. The other produces alcohol and carbon dioxide. So any questions about that slide before I show you a picture of this entire process? All right, this picture's from your old textbook. And there you go. It looks very familiar, right? We got glucose. We're breaking it down into pyruvate or pyruvic acid, as your old textbook called it. Stripping more hydrogen electrons, producing a little bit of ATP. Again, those hydrogen electrons have nowhere to go because there's no oxygen, so they're recycled and put back into the pyruvate. But in this case, we lose a carbon for that pyruvate, and we have ethyl alcohol instead and a little bit of carbon dioxide. But again, you don't need to know all those details. What you need to know is there's two different types of fermentation. One produces lactic acid, and the other produces alcohol and carbon dioxide. Any questions? All right. This was in your textbook, so I'm bringing it up, but I'm not going to test you on it. Put a big old X through this. It's an indication because I teach out of the textbook. So some prokaryotes, like bacteria and stuff, they can use anaerobic respiration, obviously. And there's different types. You can see here, there's some archaea that can reduce carbon dioxide and methane while oxidizing the NADH. But again, this is all stuff I'm not going to ask you about. The point is, microorganisms can live off of fermentation. Actually, your book gets into more details. And again, let me put a big X to this, but I'm not going to ask you. So basically, like I said, there are some microorganisms, such as bacteria, that can live off of fermentation alone. And some of them can do both. They can do fermentation or they can do regular cellular respiration, well, aerobic cellular respiration. And some of them are unable to survive in the presence of oxygen. So again, as far as this semester is concerned, and as far as this exam is concerned, this is a very unimportant slide. But if you're interested, you can look that up for an independent work. These bacteria that live where they can't have oxygen, where do they live? Where would they be? Where they have absolutely no oxygen. Anyway, any questions about this slide? All right, in summary here, fermentation characteristics. All fermentation produces gas. Well, I shouldn't even like that. Again, this is coming out of your textbook. Yeah, I'll put it next to this. But yeah, this is important here. I mean, not that's going to be a test question. Just keep that in mind, right? The purpose here is we need the NAD plus, right? That's why we just need a place to dump those iron electrons. It's always back to the pyruvate. and it either produces lactic acid or ethanol. So anyway, any questions about that? All right, let's wrap this up. We're almost done with this chapter anyway. Connections to other metabolic pathways. So it's a good time to refresh your memory. There it is. What is the very first molecule that we deal with respiration or even fermentation? What is the molecule that we're breaking down to make ATP? Definitely need to know this for the exam. I keep saying it over and over. We keep talking about it. What molecule do we break down to make ATP? Uh-oh. You guys just afraid to get the wrong answer? Surely you guys know it. You know it. What molecule? All right, we'll start, like I said, from the beginning, from glycolysis. What is that molecule that we break in half to make pyruvate? So important. Big picture stuff here. Yes, thank you. Because the whole next chapter is basically how do we make glucose. Yeah. So glucose. We keep talking about glucose, right? You break down a long molecule of glucose, you get about 32 ATP, right? We talk about glucose. How many of you had breakfast today? Just one, huh? Well, okay, then how many of you had dinner last night? All right. And did you guys eat glucose for dinner? Like, did you have a bowl of glucose? Or grill up some glucose? No, right? And you probably actually didn't have any glucose molecules, unless they were carbohydrates, but they were bound together with something, right? Point being, we eat other stuff other than glucose. So even though we keep saying glucose, we all know that when we do eat other things. So that's what we're about to talk about here in a second. The learning objectives of this little section is, by the end of the section, you will be able to discuss the way in which carbohydrate metabolic pathways, glycolysis, and the citric acid cycle interrelate with protein and lipid metabolic pathways. In other words, you should be able to understand that you do eat other things and what happens when you eat other things. I'll go ahead and tell you now, none of this that I'm about to tell you will be on the exam. It's just for your own knowledge. It's in the textbook, so I'm talking about it. So yes, respiration is versatile, right? You can eat other things, right? When you eat carbohydrates, when you eat proteins, right? It's broken down and it can be used to make ATP. That's the purpose of what we're saying here. Here's a picture that's not from your textbook. The one from your textbook is not as good, I don't think. Yeah, there you go, right? So if you eat fats, for example, depending on, when those fats are broken down, remember, fats are triglycerides. You got the glycerol molecule, the 3 fatty acids. So the glycerol molecule goes into glycolysis. The fatty acid molecules go a little bit before, between glycolysis and citric acid cycle. When you eat proteins, remember those amino acids. There's so many different amino acids, right? They're very different. So depending on which amino acid, depends on how they're being digested. I'll put a big old X through this because you don't need to know this. If anything, I might give you a picture like this and then ask you a question like, how are fats metabolized? And I would give you this picture and you would look it up and say, oh yeah, fats are broken down into glycerol and fatty acids, one of which goes into glycolysis and the other one goes into citric acid cycle. So any questions about that? All right, the next word for attendance is sushi. And that's it for this chapter. I'm going to jump into the next chapter. I'm going to try something different with this next chapter. Slightly different. Because I've read the textbook, and what your textbook has versus what the old textbook has for photosynthesis is nearly the same. So I'm going to use my old PowerPoint from the last textbook and teach you from that. I can pull it up. Are there any questions while I'm pulling this up? All right. Again, this is same information, but it's coming from the last textbook. Obviously, for us, this is not chapter 7, but it is about photosynthesis. Skip this page. There we go. Let's talk about the basics. And this is also information that's in your textbook. It's kind of in a different order. You do need to know this word, not necessarily for this exam, but later in the semester, these words are going to come back. This is your first introduction to them. I mentioned it when we first started the last chapter, but here you are again. Photoautotrophs. You need to know what those are later. Basically, anything that does photosynthesis is a photoautotroph. Plants, algae, some bacteria, anything that does photosynthesis is called a photoautotroph. Pretty simple, right? What does photosynthesis do? It transforms light energy into chemical energy. I'm sure you already knew that. Not that you need to know anything on this slide or this bullet point, but your old textbook pointed out that that chemical energy, that transformation from light energy to chemical energy, gives us stuff like food and clothing and wood and energy, right? I'm not going to ask you that. So here's some pictures, some examples of plants, algae, and some bacteria. Again, not from your textbook. I'm sure you guys all know what plants and algae look like. Here's some stuff you do need to know. Again, someone's going to miss this question. It's going to make me sad. I always ask this question because I think it's an easy question. One question is going to say blank happens, or excuse me, it's going to say something like respiration happens in blank and photosynthesis happens in blank. So respiration, the thing we just finished talking about, happens in mitochondria. Photosynthesis happens in chloroplasts. You need to know that. That is going to be a test question. It's supposed to be an easy test question. It'll be worth 2 points because there's going to be 2 blanks in it. And someone's going to miss it. But yes, you need to know that. So let's talk about what chloroplasts are. Obviously, like I said, this is where photosynthesis happens, and they are light-absorbing organelles. And they're found mostly in the interior cells of leaves. I'm not going to ask you any of these questions. The only thing you need to know is that right there. Right? You need to know this is where photosynthesis happens. So who knows what chlorophyll is? We know what chloroplasts are. I just told you that, but what is chlorophyll? Any guesses? All right, I'm going to eat an easier question. Anybody know what color chlorophyll is? Good, yes. Chlorophyll is just a green pigment. That's it. I'm not going to ask you any questions about chlorophyll, but to understand what I'm about to teach you, you just need to know what chlorophyll is. And like I said, it's a green pigment. What you might not know is a pigment is technically a light-absorbing molecule. Right? It absorbs light energy. So yeah, again, for the lab that we have this on Wednesday, I'll remind you, the reason it's green is because it is absorbing all the colors except the color green. That's being reflected. That's coming back to your eyeballs. So green is the one color that chlorophyll is not absorbing. It's like my shirt. It's blue because it's absorbing all the colors other than blue. Blue is being bounced off of my shirt and hitting your eyeballs, and your brain perceives that as the color blue. Here's another thing I'm not going to ask you about, but it's definitely in your definitely the old textbook, and I think the new one too. But stomata, right? So leaves have little holes. You can almost think of it as a mouth, right? That's where the gas exchange happens. I'm not going to ask you about that. You don't necessarily need to know it to understand photosynthesis. I guess the point is, plants do have to have gas exchange, right? Because if we're producing, if we're using carbon dioxide and producing oxygen, obviously we need a way for those gases to come in and out, and that's where it happens. I'm not going to ask you anything about that. I'm also not going to necessarily ask you anything about this, but there's something called thylakoids, and these are the membranes inside the chloroplast. So the same way when I was talking about mitochondria, and I said, oh, look how wrinkly it is because there's a lot of surface area, because there's a lot of proteins for the electron transport chain. Same situation here. Not that I'm going to ask you. But let's look at a picture. Here's a leaf. We're going to zoom in, right? If you look at this, zoom into that little square. That's what the leaf looks like. You can see that there's cells here and a vein of water, the stomata where the gas exchange happens. Then we zoom in even more, right? And now we're looking at one plant cell, right? Here's a bunch of plant cells. This is one plant cell. That's an actual picture of one. And then we even zoom in a little bit closer to the chloroplast, and that's what a chloroplast looks like. Again, not that I'm going to ask you, but that's what a chloroplast looks like. So any questions about that slide? All right. So just like when we talked about respiration, I gave you the overview, and then I went into details. So I'll do the same thing here. Let's talk about the big picture, the overview of photosynthesis. And again, everyone's different, but I recommend studying this the same way I recommend studying respiration. Look at the big picture and then start adding details. So the first thing you should memorize is this here at the bottom. Memorize this, and then you can start adding the details. You need to know that in respiration, use carbon dioxide and water to make glucose, and oxygen is a byproduct of that. Another thing to keep in the back of your mind, this is essentially the opposite of cellular respiration. Remember, in cellular respiration, we break down glucose in the presence of oxygen to make ATP, and we have carbon dioxide in water as a byproduct. And remember, like I said, in respiration, the purpose of breaking down that glucose is to strip away those high energy electrons, right? And here, it's the opposite. Here, we're taking regular old electrons and boosting them, so to speak. We're making them high energy, and we're incorporating them into a glucose molecule. So keep that in the back of your mind as we talk about this. Also keep in the back of your mind the way the exam's going to go. Like I said, it's going to be very similar to cellular respiration. In that, the question is going to say, in what stage is carbon dioxide used? In which stage is water used? In which stage is light energy used? In which stage is glucose produced? And in what stage is oxygen produced? So again, memorize that and then start adding the details. Any questions about this slide? All right, here's everything I just said in writing, right? So again, in cellular respiration, which is the thing we just finished talking about, it's a fall of electrons, right? We have those high energy electrons that start with glucose. You break it down, and all those electrons eventually go to oxygen, and that forms water. And the purpose of that was to make ATP, and we produced carbon dioxide as a byproduct. And again, photosynthesis is the opposite. So another studying hint, and you don't have to listen because everyone learns differently. But for me, I would recommend learning respiration. Have that memorized before you start studying photosynthesis, because again, it's the opposite. So technically, photosynthesis should be easier to learn once you've learned respiration, because you just think, all right, it's the opposite. And what do I mean by the opposite? Again, we have regular old electrons, but we turn them into high energy electrons. Like we boost their energy levels. That's what the sun's for. You incorporate that into glucose. So the same way in respiration, right? Electrons are accepted by oxygen, and that forms water. Here it's the opposite. Here the water is donating those electrons, and that produces oxygen. So I remember saying that in the last chapter. You should associate oxygen and water and electrons together. Whether you're talking about photosynthesis or respiration, those things go together. Again, in respiration, the oxygen is accepting the electrons, forming water. In photosynthesis, the water is donating the electrons, forming oxygen. But either way, you should associate those things together. Anyway, any questions about this slide? And again, here's everything we just said earlier. There's a picture of it just to remind you that is respiration, not photosynthesis. Which you could almost think of it as the opposite, right? You could almost just draw the arrows the other way. We have water, it's donating electrons, which produces oxygen. Those electrons go uphill, so to speak. They absorb energy, we make them high energy. And then that is incorporated into glucose or food, right? Whatever food you're eating. Any questions about that? All right, so like I said, when you're studying this, I recommend memorizing the chemical formula first. And then once you have that memorized, I recommend memorizing this fact. There are two stages. Light reactions and Calvin cycle. Most of the exam is going to be, at which stage does this happen? At which stage is this produced? Those are going to be the two answers. I'll go ahead and tell you now, the multiple choice for photosynthesis, it's a multiple choice exam. The only two answers are going to be light reactions, Calvin cycle, or both. But there are going to be other options because it's multiple choice. So if you choose something other than those two, you're going to be wrong for photosynthesis. Don't let that trick you. I mean, it is there to trick you, but I'm telling you right now, it's going to be there. So for example, it might say, in which of the two stages is carbon dioxide used in photosynthesis? And then one of the options might be glycolysis, which is completely wrong, right? Because glycolysis isn't even part of photosynthesis. So if you get those questions wrong, you'll lose more points. You'll lose fewer points, at least if you guess one of the two correct stages. Make sense? All right. Let me ask you this. What do you think? If you, based on these names, based on what I just told you about chlorophyll, which one of these two stages do you think chlorophyll is used? Think about what I just said chlorophyll is. right? So it's a pigment. So what do you think? If you just had to guess based on nothing else, which one of those two do you think chlorophyll is being used?
Light reactions.
Good. That is correct, and you need to know that. Chlorophyll is used in light reactions, which makes sense. Because remember, a pigment is something that absorbs solar energy, right? Therefore, that would make sense that the light reactions, because it's a pigment, so it's all about light. So yeah, that'll be a test question. Which of the two stages uses chlorophyll? Light reaction. And here's another test question. Which of the two stages produces, and I'm going to talk about how this happens, but for now, just know that this happens. Which of the two stages produces ATP and produces basically high energy electrons? And the answer is light reactions. And there's the other one I told you about. So when I first introduced you to NADH, I said on this exam, there's going to be there's going to be a few molecules, and they all look the same. And as far as we're concerned, they are all the same, right? So we had NADH, we had FADH2, respiration, and here's finally the third one, NADPH. As far as we're concerned, the same thing. Those things are just moving around high energy electrons. And this should make sense too. So when you get this test question, someone's going to miss it, but I hope not. Think about it. What is light? Light is energy, right? Light is energy. So it would make sense that we are producing energy, so to speak, right? ATP is energy. NADPH, that's high energy electrons. So you need to know that. Which of the two stages produces ATP? Which of the two stages produces high energy electrons? And also, let me take another step back. Big picture here. Calvin cycle, right? Correct. Big picture. Remember, the purpose of this is to produce glucose, right? That is the purpose of respiration. So what's it doing it? What's actually doing it is the Calvin cycle. Calvin cycle is the thing that's producing the glucose. It's like a little factory, right? It's producing glucose. What do you need in a factory? You need things, right? You need materials and you need energy. So light reaction is all about getting that energy. Then we'll talk about it in a second. The Calvin cycle is using that energy to produce what we're after. Another test question is right here. Which of the two stages uses water? The answer is light reactions. Because again, the water is the thing that's providing those electrons. They're going to get boosted uphill and then eventually incorporated into a molecule of glucose. And again, like I said, whether you're talking about photosynthesis or respiration, water and oxygen, you should associate those things together. Because in this case, water, once it donates those electrons, produces oxygen. A lot of test questions there. Which of the two stages uses chlorophyll? Light reactions. Which of the two stages produces ATP, high energy electrons? Light reactions. Which of the two stages uses water? Light reactions. Which of the two stages produces oxygen? Light reactions. So any questions about this side? Remember, this is still just an overview. We're going to go into more detail for all this. All right, the next word for attendance, cycle. And let's talk about the Calvin cycle. Like I said, the first one, light reactions, that's all about energy, right? Because meat and potatoes, is the Calvin cycle. That's actually doing the thing that we're after. That's the thing that's making the glucose. So the test questions are there. Which of the two stages uses carbon dioxide? Calvin cycle. Which of the two stages uses ATP, not produces? Remember, light reactions produced it. Calvin cycle uses the ATP. It needs energy to build that molecule. It needs high energy electrons to incorporate into that molecule of glucose, right? So again, which of the two stages uses high energy electrons? Calvin cycle. And this part should be easy because, like I said, when we were talking about respiration, whether we're talking about photosynthesis or respiration, you should associate carbon dioxide with a cycle, right? So citric acid cycle for respiration, Calvin cycle for photosynthesis. Either way, it's a cycle. It should be easy to remember because CO2 starts with a C. Calvin cycle, C, CO2. Citric acid cycle, all those C's, right? Hope that'll help you remember. So any questions about this side before I show you a picture of the overview? All right, it's a picture not from your textbook, but it's It's a good one. Again, we have the chloroplast, or the light reactions, right? It's taking the water and is donating the electrons. Those electrons are excited by the light, right? In this process of donating the electrons, we have oxygen as a byproduct. But the main important thing here, if we're talking about light reactions, is we're trying to produce some energy, so to speak, right? Trying to make that ATP. We're trying to get those high energy electrons because then those things go into the Calvin cycle, and that gives your cells or their cells the energy to squish all those carbons together from carbon dioxide to produce glucose. So that's it, big picture stuff right there. Again, test questions. Which of the two stages uses light reactions? Hopefully no one missed that. It's in the name, right? Light reactions. Which of the two stages uses chlorophyll? Light reactions. Which of the two stages uses water? Light reactions. Which of the two stages produces oxygen? Light reactions. Which of the two stages produces ATP and high energy electrons? Light reactions. Which of the two stages uses them? Calvin cycle. Which of the two stages uses carbon dioxide? Calvin cycle. And then finally, which of the two stages produces the glucose, which is what we're after? And the answer is the Calvin cycle. So that's it, big picture stuff. Memorize this before you start studying what I'm about to teach you. Now we're going to go into detail, like how is the light used? How is this ATP made? How are we getting those high energy electrons? We're about to talk about that. So any questions about this picture? All right. If you download the PowerPoint, you watch this little video, kind of shows like an animation of chlorophyll, or excuse me, photosynthesis. Speaking of videos, if you look at this week's lab, there's 2 videos in the pre-lab that you don't have to watch, but I highly recommend them. Some short Hank videos. He talks about respiration and photosynthesis. Does a great job. It's very succinct, very short, but to the point, and I recommend watching it. Anyway, I'm not going to ask you this. This is from your old textbook. But the incorporation of carbon from air into inorganic compounds is called, excuse me, organic, not inorganic. It's called carbon fixation. That would actually be important for Bio 108. But for this class, it's not so. Basically, again, like I said, carbon dioxide is used, right? We use carbon dioxide to get those carbons, to squish them together, so to speak, to make glucose. So obviously, anything that's doing photosynthesis is taking carbon dioxide out of the atmosphere, and that is a good thing for us. I mean, we do need some carbon dioxide. Without it, the world would be too cold, we'd be frozen. There would be no life as we know it without that greenhouse effect. But as we also know, too much of it's not a good thing. So it gets too hot. Anyway, any questions about this? All right, now we're going to get into the nitty gritty. We're talking about how things actually work. Before we get into the light reactions in the Calvin cycle, we'll give you a little bit of background information here. Stuff you need to understand to truly understand how those two things work. So let's talk about the nature of sunlight. I'm not going to ask you this, but sunlight is a type of energy or radiation called electromagnetic energy, right? So all the sunlight that you see coming from there, that's electromagnetic energy. All the stuff that you're protecting your skin from when you wear sunblock, UV radiated, UV, that's electromagnetic energy. The radio, when you're listening to the radio, electromagnetic energy. When you get x-rays, when you microwave something, that is all electromagnetic energy. Another thing you don't necessarily, I'm not going to ask you any of this either, but energy acts as waves. This is a physics class, we can talk a lot more about that. It's not, I think basically, I mean, you don't need to know this for the exam, but just for your own knowledge, the higher it is on this end, the more energy it has. The lower it is on that end, the less energy it has. So red is a low energy molecule or color. Purple is a high energy color. And that energy is based on the wavelength. And again, I'm not going to ask you any of that. You could even brain dump that, and you'd be fine. You can still understand what I'm about to teach you about photosynthesis. So any questions about this slide? All right. As you're going to find out in lab this week, when light shines on a pigment, certain wavelengths are absorbed and disappear, and the other is reflected. So like I've already said, the reason chlorophyll is green is because it's absorbing all the colors other than green. Green is the one color that's being reflected back to your eyeballs. And your old textbook points out that there was some botanists that did some experiments to figure that out. But I'm not going to ask you that either. But yes, you should know the green, in theory, is the worst color for photosynthesis for that reason, right? It's the one color that it's being reflected, one color that it's not absorbing. So any questions about this slide? I'm really going to skip through this because again, this is from your last textbook. Every chapter they had something called the process of science, and they talked about a real life experiment. Skipping through that, certainly not going to ask you any questions about it. And it's basically a different version of one of the experiments you're going to do in lab. All right, here we go. Chloroplast pigments. Again, I'm beating a dead horse here. I've said this over and over, but here it is again. Now you can read it. It's in writing. Green. Chlorophyll is green because that's the color that's being reflected. Also, your book points out that energy cannot be, or reminds you, energy can't be created or destroyed, right? So once absorbed, that energy has to be converted to other forms, and that's what we're going to talk about shortly. Another thing I don't know if I'm going to put a question mark. I'm not sure I'm going to ask you this, but it's not just chlorophyll. Well, first of all, chlorophyll itself, there's two different types. There's chlorophyll A and there's chlorophyll B. Then there's even another type of pigments. We call them carotenoids. I'm probably not going to ask you this, but let's talk about it anyway. What does chlorophyll A do? What's the difference between the two? Chlorophyll A participates directly in the light reactions. So I might ask you that, but I'm definitely not going to ask you this second bullet point. But for your knowledge, it absorbs mainly blue, violet, and red light. Right? So those different ends of the spectrum. Doesn't do very good with the colors in the middle. Anyway, any questions about chlorophyll A? All right, chlorophyll B. It absorbs mainly blue and orange light. So similar, right? Some red and orange, they're close. But it conveys the absorbed energy to chlorophyll A. So chlorophyll B absorbs some energy, right? Some photons, as we're about to learn. And then that energy is then transferred to chlorophyll A, which is where the magic happens, so to speak. But again, if anything, you need to know this bullet point. You do not need to know what color it absorbs. Any questions about chlorophyll A or B? Again, if I ask anything, you just need to know basically chlorophyll A, that's the moneymaker, right? That's the thing that's doing the work. Chlorophyll B is just there to help chlorophyll A. Then we have the carotenoids. They absorb mainly blue-green light, and they absorb and dissipate excessive light energy that might damage chlorophyll. So in a way, carotenoids are like the sunblock of the plant cells. And also, this goes back to, think about this lab that we're having. So the first experiment for this lab is, does light intensity affect respiration? I think most people guessed yes, and that makes sense, right? They're saying most people hypothesize that the more light you have, the more photosynthesis you have. And that is correct to a point, because here, the fact that we have carotenoids in these cells tells you that too much light energy is not good, right? There is such a thing as too much. Anyway, any questions about these three things? All right, your old textbook went into more detail about these carotenoids. I'm going to do it really quickly. Slightly interesting. I'm not going to ask any questions about it. The two examples are beta carotene and lycopene. Beta carotene is the thing that gives stuff an orange color, right? So pumpkins, oranges, right? Carrots, things like that. And that's important for your health because that's where you get vitamin A from. And then lycopene, we should associate that with red stuff. Anyway, again, I'm not going to ask you any questions about this, but do you have any questions about this? All right. Well, this is not very specific. So if you want to, for independent work, you can look more into this. Like, how does your body even use vitamin A? How does your body use antioxidants? Things like that. The old textbook also pointed this out. It's almost perfect timing for this discussion. Almost, but not quite. When you see the fall colors, that is from the carotenoids, right? They're there year round. Those fall colors are in the leaves year round. We just can't see them because the chlorophyll's there. Then around fall time, that chlorophyll disappears, and we're left with the beautiful fall colors. Your old textbook pointed out that it's a decreasing temperatures that causes to happen. But that, again, it's a very broad statement. So if you want to, for independent work, you can look more into that. How does it happen? What exactly causes the chlorophyll to leave the leaves? Other than decreasing temperatures, right? Like how much, how cold does it need to get? It's the time period. What happens to the chlorophyll? Can't just disappear, right? Where does it go? Anyway, any questions about that slide? All right, this slide too is a bunch of stuff I'm not going to ask you. But this shows you, right, what colors are being absorbed. Chlorophyll A is absorbing those colors. Chlorophyll B is absorbing these colors. The carotenoids are absorbing those colors. And again, you can see right here, green and even yellow a little bit, not good colors. Not good colors for photosynthesis because those are the colors that are not being absorbed. Any questions about that slide? All right, let's talk about how photosystems harvest light energy. So I've already said in the overview that the light reactions are the things that are producing the ATP and the high energy electrons. And I said, obviously, the light reactions use light, but let's talk about how that happens. Again, I'm not going to ask you this, but if this was a physics class, we would talk about this. Light behaves as waves, but it also behaves as discrete packets of energy. So here's a word you definitely need to know. Not necessarily because I'm going to ask, but because I'm going to use this word. A photon. It's a fixed quantity of light energy. So again, using my shirt as an example, my shirt looks blue to you because there's photons hitting it. And it's absorbing all the colors other than blue. And then those photons are being bounced back to your eyeballs. And your brain interprets it as the color. And again, not that I'm going to ask you this, but like I said in the previous slide, shorter wavelength means there's more energy in the photon. So blue colors, violet colors, more energy than red, for example. Any questions on what a photon is? All right, how many of you have been outside on a summer day and maybe wearing like a black shirt and you get hot? That happened to you, right? So do you know why that happens? Here's the slide to tell you. When the pigment molecule absorbs a photon, those pigments, the electrons in those pigments gain energy. They become excited. And what happens is, once they become excited, they can't stay that way. It's a long physics discussion we can't get into. But they get excited. It's an unstable state, so then they lose their excitement, right? It comes back down. But energy can't be created or destroyed, right? So once those electrons get excited, then they go back down and release the energy. That is then usually released as heat. And that's why you get hot when you wear a black shirt on a summer day. Because black is absorbing all the photons, all the colors. So all those electrons get excited. It's unstable. They go back down. They release that energy. And in that situation, it's released as heat. That's what makes you hot. Obviously, it's going to be a different story once we talk about photosynthesis. Because we're not after heat energy, right? Photosynthesis isn't about heat energy. In photosynthesis, we need ATP. We need high energy electrons so we can make glucose. Anyway, any questions about the slide? All right, here's a picture that basically shows what I'm talking about. Again, photon, in this case, we'll talk about chlorophyll. Photon hits the chlorophyll. The electrons in that molecule get excited, but then they can't stay that way because it's unstable. So they release their excitement. Yeah, that's correct. And then again, in my situation or my explanation, we talk about heat, but also your old textbook points out that sometimes that actually produces light. Like I'm sure you guys have seen this, glow sticks. The reason that's happening is because you break them, the chemicals mix, those electrons get excited, they can't stay that way. So as they lose that excitement, instead of producing heat in that situation, they produce light. Obviously, I'm not going to ask you anything about glow sticks. But again, the idea here is that these electrons get excited. They can't stay that way, and they give off that energy, right? They release that energy. And where this is important is this word right here, photosystems. So inside the thylakoid membranes, we have these chlorophyll molecules that are organized together with other stuff. Those are the photosystems. So now we're being more specific. So now I can say, where did the light reactions take place? The correct answer is chloroplast, but an even more correct answer is the photosystems, right? Because these are specific places on the chloroplast where this happens. And I'm probably not going to ask you what a photosystem is. I'm going to use that word so you need to understand what it is when I use it. Here's a picture of what I'm talking about. Again, not from the textbook. Here we're looking at a gigantic, or not gigantic, we're looking at a big chloroplast. We're going to zoom in into the thylakoid membrane. And there you go. Embedded in the membrane, you have these photosystems. You can see, it's a membrane, just like our cell membrane. It's phospholipid bilayer, proteins embedded in there. I guess we can leave it at that. So the last word for today will be cluster. And that's it. And hopefully we'll probably finish this. I think we might be able to finish this chapter up on Wednesday. And then, right now, it looks like the exam should be on Friday if it's ready. You guys have a good week.